Well, first, I would like to thank the organizer, Jérôme Delporte in particular, for inviting me to this conference. This is obviously out of my league and out of my expertise, so it's quite a challenge, but I'll take on that challenge. And also thank you for this, making me discover Besançon. Whom I didn't know the city, and it's quite beautiful, so I envy you to have a few days to spend here. Okay, so how does the human brain make sense of time? I will obviously not give you the answer. I will give you some of the questions, hopefully some questions that you can perhaps have ideas about. So the first thing that we could ask is, what's with this word time? It turns out to be a word that is being used a lot in different languages. In some languages, it's one of the first to be used, sort of in terms of frequency. It's about among the first 100 to be used. But then you could also ask human participants what they think about time. So when I say time, what do you think about? Of course, here we have an expert audience, so you're going to be very precise and have very specific things to say about it. But in fact, if you ask this to anyone and to the average John Doe, so to speak, we will realize that most of the vocabulary that we are using, the reason we're using time, sorry, I don't know if you can see, most of the associations are actually about quantifications. So you can see watch, weight, and so on, stopwatch, measure, and this is actual data. So this is when you ask people to associate with the word time. Now, what I'm interested in is not time as a physical entity, so to speak, but how the brain represents time. And that's, of course, a old question. So I'm citing here someone who is a little less known than René Descartes, but who was at about the same time as René Descartes, and who said this very nice sentence, which is translated here in a politically correct manner. The original French sentence is a little bit more critical about Locke's approach to perception. But in any case, what is suggested here is that if we do have, as humans or even animals, the ability to perceive time, then it means that the system that underlies that capacity to experience time must have some form of time measurement device. But it's not so straightforward. So I'm going to spend a bit of time to detail right away my take-home message, because it will be carried through the empirical data that I would like to show you. And the idea here is that our perception of time as human is not really linearly related to what we may think about with respect to time in physics. So basically the problem that we're confronted with is that in some ways we have a physical world in which we have a clear at our scale time dimension, the Newtonian time dimension. With very sophisticated techniques nowadays, we can have access to the dynamics generated by the brain at the scale of the whole brain. So I will talk and show you some data about this. And on the other hand, we have the mental aspect, the mental stuff, namely the fact that we can experience time, but I can also talk about time, right? The fact that I have some form of conceptual abstraction about what it means to think about time. So it's not just time itself. So what is the relationship between these three? Another quote, which is often used in the literature, is that by Saint Augustine, 400 AD. There be three times, a present of things past, a present of things present, and a present of things future. For these three do exist in some sort in the soul, but otherwhere do I not see them. Present of things past, memory, present of things present, sight, present of things future, expectation. And I guess the question in terms of the intuition that St. Augustine is raising here is whether was he speaking about time as a physical entity out there, as a physical dimension, or is he thinking here about the psychology of time? And I think he has a very good definition of the psychology of time, because a complex system that is the brain, that makes us rise to conscious awareness of the world, is always in the present, so to speak, right? We can look at it in the physical present, so to speak. But this same system is capable of making us think about the past or making us think about the future when we have to plan in a calendar and so on. So I think it's a good definition for psychological time. And so the idea here is that we have to understand which temporal structures is the brain building in these very complex dynamics that enables us to understand the world and to understand itself as well, since that's what the major problem of neuroscientists is we're trying to understand ourselves, right? So the main thing that I'd like to highlight, without going into much details, is simply to always keep in mind that we have an impoverished vocabulary when it comes to time, and we have to always be very explicit as to whether we look at it from the dimension perspective or whether we look at it as a measurement. And it is also true in terms of the representation that the brain may make of time. So if we talk about a dimension, it means that we have to have in the brain some form of stable referential system onto which information is going to be mapped. And on the other hand, if we talk about it as a measurement, it means we have some form of metrics that enable us, for instance, to perceive duration or to perceive simultaneity, order, and so on. So in my field, most of the time we hear about time as a function of physical time. So we have a sort of a taxonomy of behaviors that is entirely and linearly matched onto the timescales that we could think of, from microseconds to days and hours. And I think there's a bit of an issue with this because, for instance, when the brain actually represents the acoustic space, so trying to localize a sound source on the left or on the right, there are neurons in the brain that are coincidence detectors capable of measuring the left versus right difference between the acoustic sound source arriving to the left and the right ear. So in other words, microseconds delays in the acoustic domain can help build a spatial representation of the world for humans, for mammals, and also for bats. In other words, timing information in the external world may not raise to our conscious awareness as time itself, but also as space. So it's a bit more complicated. And so much so, in fact, that you can create illusions so that if you can do this at home, you play some sounds in the left and in the right. They are triggered. There are simple clicks, a series of clicks, as you can see here. And you displace them by a few microseconds. So for instance, here 0.7 milliseconds. And you will have a virtual acoustic source. You will start having the impression of a moving source going from left to right. And in fact, you are fooling your brain because if you look at the brain responses to the illusory acoustic that you've just created, or the real change, that is a real moving source, it's quite identical. There's barely any difference in terms of the kind of response. Okay, so this is just to introduce you that the problem is a bit complicated when we talk about time. And one of the questions in the team is, when we are looking at brain activity, are we in time? Are the dynamics that we're looking at in brain activity really telling about our perception of time? And it's not a trivial issue. So to counteract this idea of taxonomy, that is mapping behaviors onto directly the different timescales in physics, so to speak, we can have another form of operationalizing the problem, which is to, on the one hand, look at time as an experience that we have. So we all experience the fact that there are different events in the world. We all experience simultaneity, temporal order, duration, and so on. Those are on the perception side of things. And these are pre-verbal. It is likely that all animals have this sort of phenomenology. And on the other hand, there is the conceptual time, the one that we are doing every day as humans, which is the verbal conceptual world in which we live, and that enables us to communicate very complex ideas. And that includes planning, scheduling, building some calendars, and mental time travel. So I'm going to hopefully have a time to show you examples of temporal order and mental time travel today. So let's embark into this. Temporal order. Some of you might be familiar. It comes from astronomy, actually. So in the 19th century, it seems that astronomers were trying to map the sky, and they realized that there were lots of disparities across people mapping the skies. And Sanford came up with this notion of personal equation, realizing that it seemed that all astronomers have this sort of individual bias in the way they were mapping the sky. So he came up with this notion of individual bias. And this has not actually been quantified, surprisingly, although it has been noted, and to some extent that's part of a reason why we came up with lots of complicated techniques nowadays. So we were interested in this phenomenon for temporal order perception. So we had, imagine here, this is a psychological experiment. You're a participant and you're being presented with a sound on the left and a visual event on the right. It's just like a beep and a flash. And your task is simply to say which of these two events came first, the one on the left or the one on the right, irrespective of whether it is a sound or a light. When you do this, what you can do in psychophysics is build what we call the psychometric curve. So here you have the percentage of time participants reported perceiving the visual event first. And here you have the measured, so the quantified physical delay independently of the brain, right? So you take measurement tools, and these were effectively, the sound and the visual event were effectively displaced, with the sound occurring first on the left when it's minus, and the visual event occurring first when it's plus. So what you have here is the performance of the subject, and what we draw is the 50% here line, which indicates that at this delay, here it turns out to be 0, participants were at chance level in responding which was first, right? Which means that they could not actually perceive order, which makes sense since the stimuli were at simultaneity physically. Now, the trick in psychology is that when you pay attention, and this is a law that is called the prior entry effect, or a law that is a psychological law which is known for the last century, from Titchener is the name of the psychologist who came up with this. Basically, when you pay attention to a stimulus, the stimulus enters your conscious awareness first, earlier than the one you don't pay attention to. So what that means on these curves, it means that if you look again at this curve here, if I ask a subject to pay attention to the visual event, the curve is going to shift to the right. And this is the point of subjective equality. So now you need the sound to be presented slightly in advance of the visual event for them to be perceived as simultaneous by the participant. And you have the reverse. If you pay attention to the sound, you have to move forward the visual event so that simultaneity is perceived first. So effectively, when you pay attention to to events, sensory events in the world, you can shift around what simultaneity means for the observer, meaning you. Now, of course, this goes against this notion of individual bias, because if you have attention, then perhaps attention is in fact a means to compensate for your individual bias. But first, we would need to know whether there's an individual bias to start with. So what we did in this study, we did lots of different experiments. Of course, we replicate what I just showed you here. And then we make participants come for 4 months. They took part in the experiment 4 times, and we measured their point of subjective simultaneity. And what you have here is each participant as a function of the actual delay, audiovisual delay. And we used very different measures each time. The main point here is that effectively these participants tend to be biased towards seeing the sound first, whereas these participants tend to be biased to see the visual event first, irrespective of the attentional modulation we did. Meaning that each of us have a sort of a different perception of what simultaneity means between audition and vision. And with respect to attention, the interesting part is, of course, that, so we don't really show here, but in between the red dot and the blue dot, you have the extent to which attention can modulate your perception of simultaneity. And what you can see is, of course, everyone can somewhat modulate, but what's interesting is that if you look across all participants, you never have all participants being aligned to what should be real simultaneity between audition and vision. Okay. So now we were interested a bit in what happens in the brain when you are in that zone where things change around. So for this, I'm going to give you a bit of information on the techniques that we are using, neuroimaging techniques. And in our field, we have a trade-off between space, time, and invasiveness. So basically, you can be very good at giving the description of the neuroanatomy of the brain using fMRI, with which we basically measure the oxygenation of the blood going through the brain, and we infer from this that these regions are most active or engaged in a particular task that we're using. This gives you a very good spatial localization, but very poor temporal resolution with roughly 1 or 2 seconds, knowing that neurons discharge at a rate of, say, 1 millisecond. So we want to have very good temporal resolution. For this, we can use MEG and EEG, which is the technique that I like, and I'll talk to you about afterwards. So with this, you're going to have the millisecond scale resolution, but you lose the spatial resolution. And then, of course, if you want the best of both worlds, you can go with invasive technique, which is sometimes what has to be happening for patients who have undergoing surgeries because of epilepsy or because of tumors, in which case we often have access to test their brain for their benefit because we want to make sure that the part of cortex that's going to be removed is not impairing them in any ways. Okay, so the technique I'm going to talk about mostly is magneto and electroencephalography. Basically, these are two techniques that are complementary, that record the electrical activity generated by the brain. And when we have electrical activity, there is a counterpart, which is the magnetic activity, so we can measure both. And since both of them are orthogonal, they will also speak to orthogonal sources in the brain. So basically, what is being shown here is a slice of the brain that is coronal, meaning in this manner here. You can see the circumvolutions of the brain, and the MEG is going to be mostly sensitive to the sulci, which are the little dips that you see here. And the EEG is going to be complementary and mostly to the gyri, mostly sensitive to the gyri. So we tend to use them both at the same time. And basically, to give you an order in terms of what we are measuring, we need tens of thousands of neurons to be synchronously activated to see 10 nanoampere meters, sorry. Now, the kind of activity we see in the brain with these non-invasive measures is basically like any physical system. You will look at the power spectrum of the activity and you will see a 1/f spectrum. And the specificity of the brain is that you do have some peaks of activity at specific frequency, which you can see also in the time domain. And they have a name, which is theta, alpha, beta, and so on. The reason I'm pointing this out is we are very familiar with these tools. We use time frequency, and I will show you how, analysis of these signals. But I think in cognitive neuroscience, we have the imperative to make clear what is the nature of the signals that we are recording from. So we're not just looking at an acoustic waveform, for instance, we're also looking at the biological reality, which we need to account for. And in fact, in the brain, there are such signals as neural oscillations, which are fluctuations of the activity, both at the level of a single cell and at the level of a population. And they cover the range of roughly what we call the infraslow, so 0.01 hertz, all the way up to 600 hertz. But the really functional scales that are most important are the one between 0.1 and roughly 150 hertz. What's interesting also is that these neural oscillations do not scale with the brain. So if you look at the rat brain or a mouse brain or a human brain or an elephant brain, and if they display neural oscillations, they are basically at the same frequency, which is extremely interesting because it may have implication for information processing across species. So I'm going to show you now a clear example of a MEG study in which we looked at this temporal order effect using a bit of a psychological trick. So the example that I often use for this is the one that all of you probably experience, which is a badly dubbed or badly synchronized movie where you start looking at the lips and hearing the person and they're not well aligned in time. They say the same thing, but they're not well aligned. And at first it's really annoying. And then after a few seconds, or perhaps minutes, you start not paying attention to this anymore and you don't realize that they are out of sync. So you can do this in the lab. That's what we call temporal recalibration, meaning that at some point the brain seemed to adapt to this disparity between audition and vision, and we were interested in this phenomenon. So to do it in the lab in a more simple way, more tractable way, we use flash and beeps again, very impoverished stimuli, very boring stimuli as well. And so the participant is going to be shown, for instance, a sound being presented 235 milliseconds before the visual event, and this is going to happen for 1 minute. So that's what we call adaptation. For 1 minute, you're being adapted with a delay. And at the end of the experiment, at the end of the block, we're going to measure again the point of subjective simultaneity, which I showed you before. And people have done this sort of paradigm before. What you observe here is that when you're going to adapt with a sound coming before the visual event, participants are going to shift slightly their curve toward being more accepting of this kind of delay. So they're going to shift their simultaneity in the direction of this delay. And in the other delay, here when you have the visual being presented before the sound, the curve is going to shift in the other direction. So basically you can play around transiently with your perception of order. So this is what we tested in the machine. This I think I can pass on. And I would like to boil down this experiment that we're doing in psychology to what it really means, because that's what we're targeting with MEG. What it really means is that in the physical world, we're presenting sensory stimuli that are 200 milliseconds apart. And they're always 200 milliseconds apart for about a minute. But in my brain, after a minute of adaptation, it's as if the auditory and the visual event were now closer in time in terms of how the brain represents these sensory events. So that's what we're trying to explain. So this is what an MEG looks like. You basically seated here in a chair. You put your head under the helmet here, which contains 300 some squid sensors. So combination of magnetometers and gradiometers. And the participants are doing the task while we record their brain activity. And this is basically the different experimental condition, which I'll skip. Another trick that we used from neuroscience is that when you present a rhythm to the brain or to a neural system, the system is going to somewhat resonate. I should use that term carefully, but basically I'm going to show you the data here. So if you systematically present a rhythm of 1 hertz, so an event happens every 1 hertz, you look in sensory cortices and the brain activity is going to also discharge or be active every 1 second or at 1 hertz. So I'm going to show you this here. So what we did here is to present the audiovisual delay at effectively 1 hertz. So this pair was desynchronized, and then the pair occurred every 1 second also. Now, if we had, in the brain, we basically, as I mentioned, have neural oscillators. So when we use EEG and MEG, we are looking at a sum or an average, group average of potential oscillators. And what you would expect in that case is that you're going to have a first event triggering a response in the brain, a second event, a stronger response, and so you start having a bigger and bigger power in that frequency response. In our case, it would be 1 hertz. And most importantly, you would expect that the locking of the brain activity would be always in phase with a stationary clock that is provided by the sensory events, right? So to make it a bit more concrete here, this is the kind of signals we observed in brain recordings using this experiment. We can do fancy source reconstruction. I won't go in it because we have to solve the inverse problem, so it's difficult, but we can do it. And so we can look at, have an idea of which are the sources that generate the signal that we're trying to understand. Here what you have over time, the 0 corresponds to the presentation of a sensory event. The red trace is the time average of the brain response to that event. And the dark traces are the single trial, so single presentation of the event in the course of the adaptation period. And what you should clearly see is that there's a 1 hertz oscillation building up. And when you look at the power spectrum, you can look in visual cortices and you see a peak at 1 hertz. In the auditory cortices, you see a peak at 1 hertz. So this is what we loosely call resonant property of the brain here. Again, I use that cautionary. Okay, so to sum up again, the working hypothesis here based on our knowledge of what these 1 hertz and entrained response or resonant response are, we would expect to have the phase either to be entirely random or to be entirely locked to the stimuli, which is this one. And our hypothesis based on this phenomenon here was that perhaps, in fact, the phase of this 1 hertz response may show us something or be telling about why it is that at some point before the adaptation we perceive this, and at the end of the adaptation we perceive this. So for this to happen, in our case where the sound occurs before the visual event, it means that the sound would have to be pushed forward in its analysis or in its representation in the brain and pushed backward in time in the visual cortex to represent visual event in the past. Okay? So for this, what we do is basically look at all of the traces that we recorded in auditory cortex and here in visual cortex. So these are inflated brain that we use often in our field. This is the front, this is the back. From each of these sources, we have plenty of signals for which we're going to derive the phase response. And we compare this across the different conditions. So we had a control condition in which the sound and the visual event were never displaced in time. And when we compared these conditions, we found at the beginning of the adaptation and at the end of the adaptation, when participants had no change of perception of physical simultaneity, there was effectively no difference in the phase response. When we perturbed the system, so when we had desynchronized audiovisual event, we had a change in the phase response. However, it didn't go fully in that way. In the hypothesis that I was telling you, we should have expected this red distribution to go up and this one to go down, but they all went in the same direction because the psychological manipulation yield, in fact, perception of participants to recalibrate in all the same directions. So there are other reasons why, which is based on the psychology that I won't go into it yet. Nevertheless, what was interesting is basically the variance in the way participants perceive simultaneity, which is the same as this individual bias, right? So we also had this here. And what we now did is look at the changes in the participants' point of subjective simultaneity, so our measure of what participants consider to be simultaneity, perceptual simultaneity, and the differences in the phase response at the beginning of the experiment and at the end of the experiment. And what we found in auditory cortex only was that the phase difference between the beginning and the adaptation period was predictive or was highly correlated, linearly correlated with participants' change in simultaneity perception. So this was quite exciting. One important part here is, of course, we are using a experiment which is which is in the lab. So we selected 1 Hertz as the frequency. So we're not claiming here that the 1 Hertz is a clock. We're just simply saying that the capacity that the brain or that the cortex has to generate neural oscillations that can synchronize with the external environment can also display some non-synchronization to some extent because you have a phase delays. So non-stationarity basically with the stationary stimuli that are being given. So I can't go in all of the details, but we have other evidence that allow us to do something like this. The way cartoonishly what it would mean here in terms of a conscious representation of time in the brain is that these are your response that are locked to the veridical time, so physical time, and the way I can, as an external observer, measure time. And then in your brain, you can create this sort of oscillatory response, which eventually would do something like this. So of course, there are some parts of the brain response that are totally mapped to our linear view of time, and then some other parts that are giving the temporal reference for the encoding of this information that can vary with respect to the external environment. So the point here is that in the field I'm working on, there's a lot of people being very interested in neural oscillations and in synchronization, the notion of synchronization. My take is that it's very interesting that we can synchronize, but at the same time, in particular as a human being, I have a mental representation of the world that is invariant with the world itself. So for instance, if you go in a room and you see a chair, you will never see that chair with exactly the same lighting, with exactly the same angle, with exactly the same color, with all the kinds of features that as a physicist, for instance, you can use to quantify that chair. And yet my brain classifies that pattern of information as a chair, independently of all the small glitches and differences in the input signals. So what that means is that at some level in the brain, we have to account for the fact that this system is somewhat related to the external world, but not fully synchronized with it, right? Otherwise, we could not have this system of representation. So that's a little bit in the overarching things that I want to say here. Do I still have 10 minutes? I'm very bad with timing, which is why I want to make sure. Okay, so I'm going to transport you very briefly into time travel, the way we conceive of it in neuroscience. I mentioned it already. We basically have a capacity to imagine the future and have conception of time that goes beyond the experiential part of time. So with Baptiste Gauthier, a postdoc in my team, we went a bit wild and asked the question whether time and space were encoded in the same way in the brain. And for this, we gave participants a map. We gave them real historical events, and we asked them to learn by heart these historical events. Some of them are past, some of them are future. So the death of Miles Davis, Ronald Reagan as president, and some of the future is one highlighted here, which is a barrage that will be constructed in India. And all of these events were mapped out in space and in time in a very proper way, which I skip. The point of this experiment was to ask participants, now that you have this list of events that are true and that have occurred or that will occur, I'm going to ask you to self-project either in time or in space and basically change your mental viewpoint. So imagine, for instance, right now you're in Besançon, but you project yourself and you're in Dubai. And now that you're in Dubai, I'm going to ask you whether the election of Ronald Reagan occurred in the east or in the west or where you are mentally. This is extremely complicated. You can do it, participants did it, and this is a case of spatial navigation with absolutely no virtual environment, right? You're just imagining and figuring out, you're thinking basically. Then another one would be you're in Dubai and for instance, so is the barrage in India going to be constructed earlier or later than where you are right now? Which is, of course, you're now in Dubai, so you can do the math. Okay, so when you do this, I'll just ask you to trust me. It's a complicated experiment, but participants are very good at doing it. And in psychology, what we measure are reaction times and your errors. So when we ask you the same question when you're now, that is not doing any mental time travel or mental space travel, you have a particular reaction time. As soon as we tell you, you go back in the past or in the future, or you go west or east or where you are now and do the exact same task, your reaction time is slowed down, meaning something happened in your brain. It's not like you're not doing the task. And then, curiously enough, your error rate decreases with the distance. So the further away in past or future or east or west events are, the better you are at dealing with these events. And this is called crowdedness. That's a property of memory in the brain. Okay, but our question was, well, is the brain responding the same way whether you're going mentally in the east or mentally in the future? In fact, it doesn't, although lots of theories in the field are claiming or have been mentioning that it might. So this time we use the fMRI because, as I mentioned earlier, it's easier to look at location or the networks when we use fMRI than when we use MEG. And what you can see here is that all of the areas that are in blue means that they are more activated when you are doing a mental space travel, and all the areas that are in red are those that are implicated in mental time travel. Now, what is very interesting here is basically by using this very impoverished signal, that is, the body is not moving in space, it's not moving in time in this experiment, you're only thinking about it, you're only trying to manipulate information along those dimensions. Then when you do this, these regions here, which are the parietal cortex of a human brain, are the regions that are highly implicated in spatial navigation, real spatial navigation. And the ones that are in red are actually not really known why they would be implicated here, because they're not part of a language system or the mathematics system. They're more executive, they're more high level, which is interesting because of course mental traveling time in the future may not be a given for all animals and might be one of the properties of a human brain. Corbalis would say so. Okay, and then I could pass on for different details, but to cut short, I just want to show you one slide which is unpublished data in our field. We looked extensively in the psychological paradigm, we looked extensively in the fMRI data, and what we were interested in is what every time researcher is interested in is the time arrow, right, the psychological time arrow. This paradigm allows us to have events that were mapped in the past, events that were mapped in the future in my brain. So we wanted an index of this, and we couldn't find it with fMRI. So we went back to MEG again to have a time resolution, and what we find is in fact a index of these ordinalities. So what you have plotted here is the amplitude response of one brain region which is encompassing the hippocampal structures, which are famous structures for memory and navigation. And the lower the activity, the more in the past you are, the higher the activity, the more in the future you are. And it correlates also with the distance. So each dot here is a different event in the historical time. Meaning that we can start having access to this, how the brain may actually construct this notion of ordinality, which is one of the most complex operations, perhaps, that the brain does. So to close up, the interest in my team is how the brain can measure but essentially represent time, because there's no linear mapping between what we call time out there and what we call time in our mental experience. And for this, and for many other things, I think we actually need really seriously to consider that the reference frames that the brain is using may not be the ones that the external observer are using. in which case we have a lot of work to do. And of course, I will thank my team. And the work I've showed you is from Anne Quessem, Laetitia Grabo, both of them were PhD, and Baptiste, who was a postdoc, is now at the PFL, and all of the different people and the funding agency. Thank you very much. Yes. 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 Yes, absolutely. Here we go. So you're talking about this here. Yes, that's extremely good insight. So when you look at the brain at rest, I think I have it better on this slide. This would be the brain, let me see here. This would be the brain at rest, or the whole system brain at rest. And even when you don't do something, and in particular if you close your eyes, you're going to see this peak of activity, which is the alpha band, which is roughly 8 to 12 hertz. And that's a historical peak of energy, so to speak, of peak of activity. That was the first that was discovered, and that's one that is used a lot in clinic to determine the states of consciousness. So attention is going to modulate that state, coma is going to modulate, sleep is going to modulate it. And that's very interesting that you point this out because it is the one that we see irrespective of what the participant does, it is considered to be an endogenous oscillator whose sources can be both distributed or very local. So it's still something that is not quite understood. And that's the preferred one in our field because it is spontaneously generated. The ones that I mentioned, the 1 hertz, is one that is effectively untrained by the external environment. So you can generate these 1 hertz, but some people dispute their nature in terms of oscillator because they are untrained by external stimuli. It's open. Yes, so in this particular experiment, no, but I know some groups who are trying to look at all of this and map out the sensitivity. Even depending on which frequency bands you are, you may be stuck to that one. In particular, the low frequency range seems to be, if you're in the theta range, you're in the theta range. But the higher up you go, the more variability you start having in terms of the peak frequency.
At the same time with a special kind of homogeneity of the system of equations that would govern this process and which probably you don't have yet put on paper, written on paper. Yeah. And that's one thing. And the second is about the diagnostic power that has been pioneered by Professor Musha in Japan. That allows, of course, to find out for a patient in coma whether he's recuperating, has a 1/f spectrum of the brain waves, or whether there is a 1/f squared agony or white spectrum of a dead person, brain dead person. Is this in fact applied, or can you tell something about the practicality of it, how it's used?
So these are complicated questions. I would say for the last 10 years or so, the field has moved forward in trying to address this 1/f. So we don't really know the origins. The complication is that we have a problem of scale. So if you look at it from the single neuron level and you look at spiking activity, then you have a whole field that has been looking at it from the avalanche, neuronal avalanches, criticality, basically, self-organized criticality. When we look from the perspective of the whole brain, like we do, we know that we have noise that is embedded in what we're measuring because we're looking at scales of thousands of neurons, so it's activity that is already based on many atoms, so to speak, biological atoms for creating that activity. The 1/f spectrum, we're starting to look at it with colleagues from the multifractality perspective to see whether it could be an interesting tool to use. And some researchers are trying to look at the correlation with task, with behavior, with various things, but it's really at the beginning. I don't actually know the work of Professor Musha you mentioned, but yeah. So that's, again, that's an open question. I should say our field is very young, right? We started really getting at it in the 1950s, 1960s. Cognitive neuroscience burgeoned then, so we are really young. Lots of unknowns.
We have time for one last question.
Extremely exciting topic, and as you said, it's quite open to various interpretation. The question that I have is the ethical aspect of entraining the natural neurological oscillation that you refer to, and whether there is any potential for creating any kind of impulsive behavior in your subjects.
So the kind of entrainment that we use is non-invasive in the sense that this is what you do every day, right? You have stimulation, you have stimuli that are coming at you. Language, for instance, is a very rhythmic, parts of it is a very rhythmic sensory event. So your brain actually locked onto this. There's a different aspect in terms of entrainment, which are techniques like TMS, TDCS, TACS, which are slightly different because they induce or they modulate brain response based on magnetic or electric signal and impulses. So, but our field is submitted to really, really stringent ethical regulations. So even when we use these tools, which are, you know, not fully non-invasive, but they are deemed non-invasive when we respect all of the threshold and so on. Yeah. So there's no risk that I know of entrainment to do the short response. There's no risk that I know of invasiveness in using entrainment paradigms.
Let's say if you have a Parkinson patient.
Oh, that's different, yeah.
Or if you have someone with the substance abuse problems.
No, so Parkinson is actually very beneficial, and you can go on YouTube, actually, that's amazing, right, to see the DBS, basically. This principle of entrainment has been shown to restore motor function in Parkinson, so avoid basically the trembling and the tremors. And if you go on YouTube, you can see that. So you see patients where DBS has been turned on and they can walk again, and turn off and they start tremoring and not being able to move. So this is a very good technique that, as you say, it has been used for OCD treatment when it's very, very untreatable otherwise, perhaps also depression. But it's a little bit like, I'm not going to be very politically correct perhaps, but it's a little bit like when you take a medicine, right? We don't necessarily fundamentally know exactly why and how it works. It works, but sometimes we just don't know how. And DBS, we don't know, we don't have a brain theory that enables and account for why it is that entrainment in that case would alleviate the symptoms. There's no fundamental understanding of it yet. It will come up. I mean, people are actively working on it. But
Thank you very much.
yeah, thank you.
So let us thank Virginie van Wassenhove again.